Hey there. Have you heard of CRISPR? It's actually a pretty cool family of DNA sequences that can be found in bacteria and archaea. The name stands for clustered regularly interspaced short palindromic repeats. These sequences come from DNA fragments of bacteriophages, which are viruses that infect bacteria. So what's the deal with these CRISPR sequences? Well, they're actually used by the bacteria and archaea to detect and destroy DNA from similar bacteriophages during subsequent infections. It's like the organisms have their own little immune system that helps them fight off viruses. Pretty neat, huh? Hey there. Let me explain to you a bit about the CRISPR array. It's basically a sequence that consists of an at-rich leader followed by short repeats separated by unique spacers. The size of the repeats is usually between 28 to 37 base pairs, and the spacers can range from 32 to 38 base pairs. Typically, a CRISPR array contains less than 50 units of the repeat spacer sequence. You might find clusters of Cas genes next to CRISPR repeat spacer arrays. There are two classes of CRISPR-Cas systems, Class 1 and Class 2. Class 1 uses a complex of multiple Cas proteins, while Class 2 uses a single large Cas protein. Class 1 is further divided into types 1, 3, and 4, and Class 2 is divided into types 2, 5, and 6. All in all, there are 6 system types, which are then divided into 19 subtypes. Hey there. Let me break down the stages of CRISPR immunity for the 3 major types of adaptive immunity in a way that's easy to understand. The first stage, called acquisition, begins when the invading DNA is recognized by Cas1 and Cas2 and is cleaved into a protospacer. This protospacer is then attached to the direct repeat that's next to the leader sequence. In the third stage, a single-strand extension repairs the CRISPR and duplicates the direct repeat. The cRNA processing and interference stages occur differently in each of the 3 major CRISPR systems. In stage 4, the primary CRISPR transcript is cleaved by Cas genes to produce cRNAs. In stage 5, type 1 systems use Cas6e Cas6f to cleave at the junction of ssRNA and dsRNA formed by hairpin loops in the direct repeat. Type 2 systems use a transactivating RNA to form dsRNA, which is cleaved by Cas9 and RNAi. Type 3 systems use a Cas6 homologue that doesn't require hairpin loops in the direct repeat for cleavage. In stage 6, secondary trimming is performed at either the 5' or 3' end to produce mature cRNAs in type 2 and type 3 systems. These mature cRNAs then associate with Cas proteins to form interference complexes in stage 7. For stage 8, type I and type 2 systems require interactions between the protein and PAM sequence to degrade invading DNA. However, type 3 systems don't require a PAM for successful degradation. In type 3A systems, base pairing occurs between the cRNA and mRNA, while in type 3B systems, the DNA is targeted instead. Overall, these stages of CRISPR immunity are crucial for bacteria and archaea to protect themselves from harmful invaders. The CRISPR genetic locus is a natural defense mechanism that helps bacteria protect themselves from repeated phage infections. Let's take a look at how it works. In the left panel, after a phage injects its DNA into the bacterial cytoplasm, a protein complex made up of Cas1 and Cas2 forms. The complex acquires a sample sequence called a protospacer from the phage DNA, which is then integrated into the CRISPR genetic locus. The sequence is now referred to as a spacer, and the CRISPR genetic locus stores several spacers from past encounters with phages. In the right panel, spacer sequences are transcribed into RNA, and Cas9 protein is synthesized. The RNA then binds to Cas9 to create a surveillance and interference complex. The complex scans the bacterial DNA for any matching sequences. If a matching phage sequence is detected, Cas9 destroys the DNA by cleaving it. This is a clever way for bacteria to protect themselves from future infections. Here's what happens in the CRISPR genetic locus, which is responsible for protecting bacteria from viruses. The CRISPR locus is made up of a few different things, including a Cas operon, a CRISPR array, and genes for transactivating cRRNA. RNA molecules called tracrRNA, Cas9 RNA, and pre-cRRNA are all transcribed from this locus. TracrRNA folds into a complex shape with 3 loops, and Cas9 RNA gets turned into protein. Pre-cRRNA is synthesized as one long strand, but it needs to be processed in order to work properly. That's where tracrRNA comes in again. It binds to certain parts of the pre-cRRNA sequence, and then Cas9 protein joins the party to form a ternary complex. This complex gets cut up into smaller pieces by enzymes called RNase-3. The final product is a surveillance and interference complex made up of Cas9 and a combination of tracrRNA and cRRNA. cRRNA has 2 important parts, the spacer sequence, which matches up with viral DNA, and a sequence that binds to tracrRNA. Together, they form a duplex that can seek out and destroy invading viruses. To execute its interference function, CRISPR-RNA must be created from the CRISPR sequence. Initially, cRRNA is transcribed as part of a long transcript that covers much of the CRISPR array. This transcript is later sliced by Cas proteins, producing cRRNAs. However, the method for generating cRRNAs varies among CRISPR-Cas systems. Type 1e and Type IF systems use Cas6e and Cas6f, respectively. These Cas proteins recognize stem loops that form when identical repeats flank the cRRNA. They cut the longer transcript at the edge of the paired region, resulting in a single cRRNA with a small leftover repeat region. Type 3 systems also use Cas6 but without the stem loops. Cleavage happens when the longer transcript wraps around the Cas6, allowing it to cut upstream of the repeat sequence. Type 2 systems do not have the Cas6 gene, so RNase-II takes over the cleavage role. These functional systems have an extra small RNA, called a transactivating cRRNA, which matches the repeat sequence. When tracrRNA and the primary CRISPR transcript are transcribed, they form double-stranded RNA at the repeat sequence. RNase-II targets the dsRNA, resulting in cRRNA production. Unlike the other 2 systems, the cRRNA is not complete, and one end of the spacer is truncated. Ribonucleotide complexes with Cas proteins form cRRNAs, which detect foreign nucleic acids. When the CRISPR-Cas system is used to fight off infections, it goes through different stages depending on the type of system being used. In Type 1 systems, the CRISPR RNA must pair up with a specific sequence on the virus DNA, known as the PAM sequence, in order to work. Once this pairing happens, a protein called Cas3 comes in to destroy the virus DNA. In Type 2 systems, the Cas9 protein is responsible for the job of destroying the virus DNA. For this to work, both the cRRNA and another RNA called tracrRNA must be present. The PAM sequence is also important in Type 2 systems, but it's recognized on the same strand as the cRRNA instead of the opposite strand like in Type 1 systems. Type 3 systems are similar to Type 1, but instead of targeting the virus DNA, they target the virus RNA. However, they have also been found to target DNA using a different protein called Cas10. In all 3 types of systems, the cRRNA contains a specific sequence that helps it recognize foreign DNA and RNA. But the system also has a way to recognize self-DNA and RNA, which prevents it from attacking the body's own DNA. CRISPR is a fascinating technology that has found numerous applications in various fields. For instance, in gene editing, scientists have utilized CRISPR to engineer probiotic cultures in the food and farming industries. It has also been used to inactivate genes in human cell lines and cells, modify crop strains, and even produce biofuels. In fact, there was a case where CRISPR was successfully used to treat a patient with both a genetic disorder and HIV disease. Excitingly, this gene editing technology might even be able to create new or revive extinct species in the future. In the field of diagnostics, CRISPR-Cas has already made significant strides. It has been used to directly detect nucleic acids with exceptional sensitivity, down to a single molecule. Additionally, researchers are actively exploring CRISPR-Cas platforms for the detection and inactivation of SARS-CoV-2, which could have a significant impact on our ability to manage and control the ongoing pandemic.
I have thought a lot about the way that the CRISPR technology might be used, and I mostly feel excited because I think that it's a very exciting technology that's going to do a lot of good in human society and for human health. I do think that there are reasons to be cautious in the way we implement it for various things, in particular for doing clinical applications in embryos, like human embryos. If you do the editing in adult tissues, those are changes that are made in an individual, but they're not passed on to children. I think most people would feel that genome editing in adults is, or at least might be, for some applications, a very appropriate technology. It might be analogous to taking a pill or any other kind of therapeutic drug that you might use in a patient for treating cancer or some other disease. And if you could do something profound like eliminate sickle cell anemia or eliminate cystic fibrosis, or have someone not have to suffer from Huntington's disease anymore or worry about getting it when they get older. What certainly raises ethical implications is whether that sort of change should be made in eggs or sperm or embryos, where those changes could then be passed on to the next generation. So I feel that it's appropriate with guidelines to conduct research in those kinds of systems, even in human embryos. I don't think that it would be appropriate to use this technology, at least today, for any kind of clinical application in human embryos. The science is going 1,000 miles an hour. One of the roles that we as scientists need to play is to really communicate the power of this technology and how we can be responsible in using it. For that conversation to carry weight globally, it really needs to be a conversation that involves scientists that are international. Do we know enough about the human genome to understand the impact of making changes to it in a developing embryo? And so I think that there I would like to see our society draw a line and say that, we won't go there right now. We'll take the time that we need to really think about this. I think the thing that's amazing about science and about biology for me is that there's always more to be discovered. And every time we feel like we've figured something out, it seems to open up several more questions. Scientists have appreciated for a long time that once we understood the DNA sequence in cells, if we had a tool that would allow easy manipulation of that sequence, that would be a very powerful kind of technology. CRISPR is a technology for changing the sequence of DNA in cells in a precise fashion to correct mutations that might otherwise cause disease. It's going to enable a lot of science to be done that was impossible to do in the past. So the way the CRISPR technology works is by the action of a protein called Cas9 that functions like a molecular scalpel for DNA. The CRISPR-Cas9 system has an amazing ability to recognize a particular DNA sequence in a cell that may be malfunctioning and then disable it by cutting the DNA. We call this gene editing, and we can use it to disable or repair a mutated part of the gene which may be causing disease. For Cas9 to find the malfunctioning DNA, we attach it to an RNA sequence that matches the DNA sequence we want to edit. Then we put this RNA-Cas9 combination into the cell. It finds the mutant DNA and uses a chemical reaction to cut the DNA strand right at the spot where it's malfunctioning. After that, we can sometimes insert the correct version of the gene for the cell to work properly again. It's a very exciting technology that's going to do a lot of good in human society and for human health. It stretches from human therapeutics to agricultural applications to thinking about how do we make better biofuels. These would be incredible outcomes, I think, of using this kind of technology in the future. So I'd like to thank you all for coming today. I'm going to tell you about a technology that is revolutionizing biology and offers the potential to cure human genetic disease. So this story really starts with DNA. You probably know that DNA is the code of life that gives cells the ability to function, gives organisms the ability to develop, but when there are mutations in that code, it can also give rise to disease. And we know a lot about the sequences of DNA in cells right now, and we know about mutations, but up until now, it hasn't been possible to do much about those mutations. Imagine that we had a tool whereby we could actually fix individual mutations in DNA, much like you would do with a word processor to cut and paste and edit text. What if we had a text editor for DNA in cells? And it turns out that now we do, and this is a technology that came about through not a focused project, but actually through a curiosity-driven set of research experiments that were done to understand how bacteria fight off a viral infection. So this was an international collaboration between my lab at the University of California at Berkeley and the lab of Emmanuel Charpentier, who at the time was doing this research in Sweden at Umeå University. That's the team. And we worked together to figure out how bacteria employ an enzyme, a protein called Cas9, that turns out to be a programmable protein. This is a protein that in nature is programmed by two separate molecules of RNA, one of which provides sequence information that allows this protein to recognize pieces of DNA that have a sequence matching the sequence of the RNA. And from that understanding, we were able to engineer this as a two-component system, a single protein and a single RNA that provides scientists with the ability to program this enzyme as a molecular scalpel to cut double-stranded DNA at sites that are directed by this piece of RNA. And once that happens, cells have ways of repairing double-stranded breaks by introducing small changes in the genome or sometimes whole new pieces of DNA that introduce new genetic information at the site of the break. So this is a precision tool that now allows us to take this protein RNA complex and introduce it into cells or tissues to correct mutations at sites where we know there's a deleterious change in the genetic code. So I wanted to show you an example of how this can be utilized. This protein complex can actually be injected directly into fertilized eggs of a mouse. And in the experiment you'll see, we're targeting a gene that is responsible for the black coat color in mice. And so normally these mice have beautiful glossy black coats. And once we make this targeted change, we then implant these edited eggs back into a female mouse. And when she gives birth to pups, you'll see that the pups are now mostly white. And the remarkable thing about this experiment is that when these mice grow up, you can test them and show that every cell in the body has this single genetic change that gives rise to the white coat color, but otherwise they're absolutely normal. So they are normal mice, no mutations elsewhere in the genome. This is a type of experiment that used to take at least a year to create a mouse like this, and now it can be done in a few weeks and by people that don't have to have special expertise. So this is a technology it's been very exciting over the last 2.5 years to see this technology taking off. These are publications in the scientific literature. It's been sort of exponential growth of publications, people using this technology for all sorts of applications. And so some of these include making changes, targeted genetic changes in plants, in fungi, in animals that are important agriculturally, in animals that are important to us as pets, and also in thinking about human health, also to do things like make changes in stem cells, which are cells that can give rise to new organs. Also to make changes in animals that are important as models of human disease, such as mice and monkeys. And we think within the not too distant future, it will be possible to actually use this technology to make changes in humans so that we can actually cure diseases that have genetic causes like cystic fibrosis and other such disorders. Thank you. In talking to my friends who are scientists, I think we all sort of feel that sense of wonder, that sense of discovery, this feeling that even the most trivial thing that I'm doing in the lab, I might be the first person ever to know this little piece of information, and it feels really interesting. It feels really fun. It's like I'm peeling the onion. I'm peeling back a little bit more of the truth. I'm trying to learn about, how is our world working?
I have a lot of ideas of different endeavors I want to pursue. I think it's my personality a little bit, always need to be on the move. I think as a scientist it's good to not always be in a comfort zone. As the first time I heard about CRISPR was around 2006, when I read some articles explaining that CRISPR could be used as a way to classify in a more, let's say, fancy manner, different strains of bacteria. So this was the first encountering with the CRISPR systems. But then I read about it and that was it for me.
I think I first heard about it from Jill Banfield, my colleague here at Berkeley, sometime around 2006. And she was working on bacterial genomes and sequencing bacterial DNA, finding lots of examples of these repetitive sequences called CRISPRs. And hadn't probably paid a lot of attention to them until some research that was published that showed that those sequences included little snippets of viral DNA. And this, was very intriguing because it suggested that bacteria were, keeping these little copies of viral sequences around for a purpose. Some research suggested that CRISPRs were using these little RNA copies of DNA sequences to detect and destroy viruses.
I was at the time a postdoctoral fellow in the lab of Jennifer Doudna, kind of fresh off the boat. This was at the time when Rodolphe Barango's science paper came out, establishing the CRISPR system as a kind of a primitive bacterial prokaryotic immune system. So my first exposure to this was at a journal club that we used to have in the Doudna group. I remember at the time we were, speculating how could this possibly work.
The knowledge was really limited, so at the time what was hypothesized is that CRISPR was a mechanism that would allow to recognize an infection with a virus and ultimately kill the viruses by targeting the genome of the viruses using protein and RNA components. So this was the hypothesis in 2006.
In Puerto Rico in the spring of 2011, I met Emmanuelle.
We were giving talks at the same session, the same CRISPR session.
She said, I've been wanting to talk to you. I'm glad to meet you in person because I've been thinking that you might like to collaborate with my lab on a project. We were talking about a protein that at the time was known as CSN1 and now is known as Cas9.
This mysterious CSN1 protein, surely we were predicting the function, but no one could show up to now how it was really working.
We had this idea that, okay, the Cas9 protein was somehow going to be cutting DNA, but how exactly it was going to do it, we didn't know at the time.
And for Jennifer Doudna was extreme, let's say, curiosity on really the details of how those proteins work.
At the end of the conversation, she said to me, I think it's going to be really fun to work with your lab on this mysterious CSN1. And just when she said that, this mysterious CSN1, I just felt this little chill, going down my back, thinking, this really is going to be a very interesting protein to study.
The collaboration ultimately went very, very fast. Involved a PhD student from my lab, Krzysztof Szylinski, and a postdoc from her lab, Martin Jinek.
Our goal was to really find out what the biochemical function or the mechanism of this Cas9 protein essentially was.
Right, somehow RNAs seem to be cutting.
What we do as biochemists is we set up systems in the laboratory and ask, in isolation, what does this particular molecule do? What is its function?
If you do the experiments with the components purified, you bring them together in a test tube and you show that it's working, then you know that you don't need anything more. It's self-sufficient. It's working like this. You don't need anything else from the cell.
We said, well, let's see if we can take that strategy using purified Cas9 protein, and we also made purified CRISPR RNA molecules, these little RNA copies of DNA sequences, and then combine them together, and then started testing to see, can it use the letters in the RNA of that CRISPR molecule to find a unique DNA sequence and cut it? And initially, the answer was no dice, nothing working.
This is a process of trial and error.
There could be some silly technical thing going on, like, our protein purification isn't any good, or somehow our sample that we're testing is just, has been destroyed in some way. Or maybe there's something missing from the reaction. There was a second type of RNA that Emmanuelle's lab had been studying as part of this CRISPR system called tracer.
Tracer RNA. It was an RNA molecule which we realized was very abundant in the bacteria. Most likely there was an interesting function. And we realized ultimately that this RNA was encoded by a piece of DNA that was in the vicinity of the DNA coding for the CRISPR system. Maybe this tracer RNA molecule has something to see with CRISPR. Maybe CRISPR needs other components. So I remember that the students of my lab were telling me, no, this is not the dogma. CRISPR needs the CRISPR proteins and the CRISPR RNAs. You don't need anything else.
But we started thinking about it and wondering, maybe it would be important to include the tracer RNA. And this was actually an experiment that was first done by Krzysztof Szylinski, the student in Emmanuelle's lab who was our collaborator on the other side of the big pond. And he did that experiment and got a beautiful answer, which was that yes, when you include the tracer RNA, the CRISPR RNA, and Cas9 together in this reconstituted, purified system, now that Cas9 protein could find a unique DNA sequence and cut it.
This was one of the milestones in that project, realizing that this tracer RNA was in fact also required for the process of cutting the DNA. Once we knew that, then of course we wanted to see what parts of the tracer RNA are needed for the process. I did a set of experiments that suggested that certain parts of the two RNA molecules were essentially dispensable, but they were dispensable in a way that would allow us to fuse the two RNAs together and in doing so reduce the complexity of the system. Just go directly to DNA cutting without having to put these two RNAs together first somehow. This then led to these experiments with these single RNA guide fusions.
Martin had made several versions that differed just in the letters of the sequence that would allow Cas9 to find a different piece of DNA.
By basically designing the sequence of the guide RNA, you could redirect it to essentially any sequence in double-stranded DNA.
So we had this thing programmed with 5 different RNAs, separate samples that would find, in principle, 5 different places in the DNA and lead to a cut. And we were in the lab looking at the result of this experiment. We're leaning over the light box, and we could see that each piece of DNA was a different size, corresponding to the length predicted based on the ability of these single guide RNAs to direct Cas9 to cut the DNA at a particular place. We had been able to engineer this thing to be different from how it is in nature and much simpler in a sense.
It was a great moment. This was going to be a programmable system that you could repurpose for other things, including genome editing.
Here was something that was truly, fascinating. First of all, just fundamentally about nature, and then thinking about how you might be able to use that in other settings and other kinds of cells was also incredibly fun, because the more you thought about it, the more ideas would come, because it was just you're clearly going to be so useful for a lot of things. So there was a very intense few weeks finishing up the last few experiments and writing up a manuscript.
And when we were writing the paper, actually, the bright days were coming in Sweden. It's very disturbing physiologically speaking, because you never see darkness, you just see like a gray sky. I would I would have a tendency to work a little bit around the clock and to not sleep a lot.
The excitement was building up. So the final weeks in this process, there was this, playing ping pong basically with the manuscript.
I would realize it's 3 a.m. in the morning because I was corresponding with California, and realizing that no, it was really time maybe to get some hours of sleep. And ultimately, the paper that was published in 2012 in Science, showing the biologists how they could harness the system, so providing them the guidelines, the recipes to use the system for their own purposes, which they did successfully, because 6 months after the publication of our paper in Science, very fast we saw publications showing that the technology was working for genome editing and even engineering in plant cells, in human cells, in mice, in different model organisms which biologists use. So this was very, very fast, actually.
It was clearly going to be a race, because if it worked, it was going to be so exciting. I wasn't thinking about, oh my gosh, this is a tool that, it fundamentally allows us to change our relationship with nature. It actually allows us to change human evolution if we want to, right? It's that profound. And it puts into all of our hands as human beings a tool that we've never had in the past the ability to change the fundamental chemical nature of who we are in this way, right? And now we do. And what do we do with that?